Okay, well, okay, I think all right. First thing we're going to do, of course, look at cell biology. Cell biology, in fact, biology in general, is a whole bunch of complex interacting chemical reactions. And we're going to go and we're going to start from the chemistry to the properties and structure and function of the molecules, major molecules of life. And then we're going to put it together to see how the cell actually works in various cellular processes. So what we're going to do first, of course, is we have to introduce some key chemical concepts. Now, once again, many of these concepts should be, and maybe even are, old hat to you folks. But we're going to introduce some terminology and some basic concepts. If it's old hat, old review to you, please forgive me, but it may be new for some people, especially ones that have not had college chemistry yet. So we're going to introduce some of these concepts and start from the bottom and go up and then start getting into things that are more important for biochemistry and molecules of life. So first of all, okay, basic unit of matter, the atom. That's the smallest unit of matter that has the properties of things. Now, atoms themselves are actually made up of a couple different kinds of components. One of them is at the center of the atom, you have the nucleus. Now, the nucleus consists of these particles, they're called subatomic particles, called protons. And protons have a positive electrical charge, and then neutrons, which have no charge. Okay, that's the nucleus of the atom. Now, orbiting around the atom, more details on that, are going to be another set of subatomic particles called electrons. Now, electrons have a negative charge. The charge on the electron is exactly equal but opposite to the charge of a proton. So you put the two together, they're electrically neutral. And in an ordinary atom, you're going to have the same number of electrons as you have protons in the nucleus. So ordinary atoms are electrically neutral overall. They've got positive charges, they've got negative charges, but they cancel each other out. Now when you look at the atom too, the protons and neutrons are about the same size, about the same mass. But the electrons are much, much less massive. In fact, it takes almost 2,000 electrons to equal the mass of a single proton or neutron. So even though the charge is the same, opposite, but the same intensity, the mass or size of the electrons are much, much smaller. Now, atoms, surprisingly, are almost entirely empty space. A typical atom is about 100 millionth of an inch across, or maybe about, say, 0.4 nanometers in the metric scale. A nanometer is a billionth or 10 to the minus 9th meter. That's the size of an atom. They're pretty small. But despite that, atoms are almost entirely empty space, because the nucleus, which contains the vast majority of the mass of an atom, is very, very tiny. It's about 10 to the minus 15th meters across. That's really, really small. If you took a golf ball and represent that as the nucleus of an atom, and went out to Legion Field or Brave Stadium and dropped that golf ball on the 50-yard line, the electrons would be circling around where the fans happen to be. That's how much empty space is in an atom. Another example. How many people have gone down to Mobile and seen the battleship Alabama? This huge thing, 50,000 tons of battleship, right? If you were to squeeze all the space out in the atoms, that battleship will be shrunk down to the size of that little battleship token in a Monopoly game. You ever seen those? They're about this long. It would still weigh 50,000 tons. You just got rid of the empty space. And of course, with that kind of mass, it just cut right through anything and go right to the center of the Earth. But that's how much space, empty space, is inside an atom. Now, as we're going to see chemical properties and chemical reactions, are dealing with the electrons. The nucleus is not involved in that. When you start doing things with the nucleus, then you're talking about nuclear reactions, all the kind of stuff that the Iranians like and Kim Jong-un likes and stuff like that. That's nuclear reaction. Oh, they love those things. They like slamming plutonium together, blowing things up with it. Okay, but anyway, atoms. Yeah, what are they doing? Okay. All right, but that's a basic structure of an atom. Now, if you happen to take and once again, in a normal atom, the number of protons, and I'll call them P+, that's a typical symbol, equals the number of electrons. So the atom is electrically neutral. That's a normal atom. That's the normal kind of atoms we deal with. Now, on the other hand, sometimes, this is very important in biology, sometimes we have a situation where you have too many or too few electrons. So number of protons does not equal number of electrons. That means that atom is going to have an overall positive or negative charges. Now these electrically charged atoms that have too many or too few electrons relative to protons, we don't call them atoms anymore. We call them ions. And ions are very important in the properties of life. For instance, calcium and magnesium ions regulate numerous things in cells. More on that later. Sodium and potassium and chloride ions are critically important in all kinds of physiological processes, ranging from muscle contraction, kidney function, water transport and xylem and sugar transport and phloem, all that kind of stuff depends on various ions. And ions in our cells and in our bodies have to be kept in a very narrow range, otherwise you're in serious trouble. Think of just one kind of ion, potassium ions. If it goes too low in your bloodstream, you go into cardiac arrest. Once in a while, it's usually around late summer and stuff like that, you hear of some perfectly healthy high school or occasionally college athlete, a football player or something like that, who collapses suddenly and dies on the practice field. Mostly because they sweated out and depleted themselves of potassium, and that stopped their heart. On the other hand, if you commit a very nasty crime in the state of Alabama, what they will do to you is pump your blood full of too much potassium, and that hits your heart and stops it dead again. That's lethal injection. Potassium is the kill agent. And almost all states do that now. I think, yeah, California got rid of the gas chamber and everybody got rid of the electric chair. So, yeah, so that's a standard death penalty in the United States is potassium. Wiping out and stopping your heart. So those ions go outside a narrow range in your history. And cells have to do things to control the relative amounts of different ions. So they pump things out, push things in, and stuff like that. Sometimes keeping concentration differences of thousands of fold compared to what's outside. Because too much or too little of these ions are lethal to the cell or to the organism. So ions are very important in all kinds of biochemical processes. All right. Now, some more terminology. I know we have to do this. Okay. Now, if we took the number of protons in the nucleus of an atom, we call that the atomic number. So every atom has a specific atomic number. Now, it also turns out that the number of protons, or the atomic number, defines the element. Now, an element is a bulk collection of a single type of atom. For instance, I have here, it's not pure gold, it's about 70 or 80% gold, but this thing here, I don't know what else is in it, but I don't know, they don't tell you. You just say how much gold is in it. Okay, this has maybe about 70% of this little thing here consists of the element gold, which is a collection of gold atoms. And gold has a certain atomic number, a certain number of protons in the nucleus. If you have carbon, carbon always has 6 protons in the nucleus. Hydrogen only has 1 proton in the nucleus. So the atomic number, the number of protons, defines what element it is and defines the properties of that element. If I take a carbon atom with 6 protons and I add another proton to it, force it into the nucleus, it's not going to be carbon anymore. It will have 7 protons. It will be nitrogen. So I could, in theory, turn carbon into nitrogen. That can actually be done in particle accelerators. You don't get much, but it can be done. It's been done numerous times. So if I change the number of protons, I'm turning one element into another. Okay, that's the atomic weight, or rather atomic number. The number of protons in the nucleus, and that tells you what atom it is. Now, we have, if you add up the number of protons, I'll just use the abbreviation, and the number of neutrons in the nucleus, we call that the atomic weight. Okay, so an element has an atomic number and an atomic weight. Now notice here for the atomic weight, we don't bother with the electrons because the electrons are too small. It doesn't make any difference. Even something like uranium with 238 protons and neutrons, those 238 electrons around there aren't even equal are barely equal a tenth of the weight of a proton or neutron. So we just forget the number of electrons when we talk about the atomic weight of something. So each element has a specific atomic number and a specific atomic weight. Now let's take a look at carbon, for instance. We look at carbon because carbon is one of the most abundant molecules in cells and in biochemistry. The term organic chemistry, which some of you guys suffered through, right? And the rest of you will suffer through. I've been there, done that myself. You suffer. Okay. The term organic chemistry at one time was referring to molecules that they thought could only be produced by living organisms. There was something magical about living organisms that could produce certain molecules that nothing else could. And then some German chemists synthesized urea, which is normally a waste product in your urine, by mixing a couple chemicals together and saying, oh, I've made an organic molecule without a life form. Okay, so now the term organic chemistry refers to chemistry of carbon-containing molecules. Whether it's made by life or not, we have plenty of organic molecules that are made in big chemical factories nowadays. But life makes all kinds of organic molecules. So, let's take a look at carbon. Now, your typical carbon atoms. Carbon always has six protons. So we could put down here, carbon 6, that's the atomic number. Now, most carbons have 6 neutrons as well, so we could put the atomic weight on top of the symbol for carbon. So that's 6 protons and 6 neutrons. But a tiny fraction of the carbon you find has 7 neutrons. It still has 6 protons. If it didn't have 6 protons, it would be something else. It would not be carbon. It would be some other kind of element. So maybe about less than 1% of the carbon you find, you have 6 protons. Otherwise it wouldn't be carbon, plus 7 neutrons. So 6 and 7 is 13, so we have carbon 13. It's a tiny fraction of the carbon atoms you find around. An even smaller fraction of the carbon atoms you have around have, of course, they got 6 protons, but they have 8 neutrons. So that's what we call carbon-14. Now the problem with carbon-14 is atoms that have a high neutron to proton ratio tend to have an unstable nucleus that breaks down, releasing a great deal of energy in the process. So carbon-14 will eventually turn into nitrogen. So carbon-14 is radioactive. Okay. Now, we have a term for these different versions of the same element that differ only by the number of neutrons they have. We call these isotopes. Isotopes are different variants of an atom that differ in the number of neutrons. They have the same number of protons, otherwise they'd be a different type of atom, but they differ in the number of neutrons. And as we can see here, most of the time when people think of isotopes, they think of Kim Jong-il and his nuclear program. They think of Fukushima Daiichi, the nuclear plant that went up last year in Japan that got flooded and leaked radiation all over the place and stuff like that. You tend to think of radioactive isotopes. Clearly not all isotopes are radioactive. Many are, but many others are not. So we have lots of stable isotopes. Carbon-14, half of the mass of that breaks down to nitrogen in a period of 5,700 years. It's a long time for us. There are other isotopes that have these, what we call half-lives on the orders of milliseconds, and still others with half-lives as long as the age of the entire universe, 10 billion years or more. Okay, so radioactive isotopes vary in how quickly they break down. All right, so clearly carbon-12 and 13 are stable isotopes. Their nucleus does not break down. They will be here. They were here from the moment of their creation in the cores of stars, and they will be here to the end of the universe itself. So they're stable, but not all ones are stable. Now, isotopes are important in biology, especially as a tool, because we can use isotopes for a number of things. Radioactive isotopes can be used for a lot of things. For instance, how did they figure out? Everybody, at least I hope everybody knows, that plants can take carbon dioxide and turn it into the sugar glucose. How do we know that?
Radioactive isotopes.
Radioactive isotopes. What somebody did was expose plants to radioactive carbon-14 containing carbon dioxide. So plants took that radioactive carbon dioxide in, and whatever they turned that into was also going to have the carbon-14 atoms in it. And lo and behold, after an hour or two, they find glucose that's radioactive, so don't sprinkle over your cereal. It's not a good thing. But that told you that plants turned the atoms that were in carbon dioxide ended up in the atoms of glucose. And then later on they found these intermediary steps. So using radioactive isotopes as tracer to find out what gets turned into what is very important in biochemistry. A lot of that work has already been done, but it still gets used. And you can use radioactive isotopes as a tag for something, where you can detect the radiation. You could attach something that would grab onto a particular protein, and it has a radioactive isotope tag on it, and then you could find where in the cell that protein is. Is it located in the nucleus? Is it located in an organelle? Is it located in the cytoplasm? What have you. And radioactive isotopes are used clinically, often for cancer treatment, radiation therapy, and stuff like that. Usually means you stuff a whole bunch of some radioactive isotope in a lead-lined container and have a little port out there. It says beam of high-power radiation comes out and hopefully shrinks or destroys the tumor. Okay, so radioactive isotopes have lots of use in both clinical purposes and as tracers and things like that to get a better understanding of location of things in cells, molecular processes and biochemistry, what gets turned into what. A lot of that biochemistry that we're going to learn, we're going to read from, was discovered by using radioactive isotopes as tracers. But non-radioactive isotopes still have some use in some aspects of biology. Now, case in point, this may become, it's looking a little bit more dubious than it did some time back, but this may become extremely relevant. Living processes. Now, the chemistry of something like carbon is all the same. You know, carbon 12, carbon 13, carbon 14. You mix it with oxygen, put a match to it, it's going to catch on fire. Well, yes, all they can take their, there you go, go to hell with it. Okay, let's see how much time we have here. Okay, all right. So, you know, the chemistry is the same. If I take carbon 12, 13, or 14 and put a match to it, it will burn, combine with oxygen and make carbon dioxide. Okay, chemistry is the same. But the lighter isotopes react just a tiny, tiny, tiny bit faster. Now, if I took carbon 14 or carbon 13, or let's use the carbon 13, I don't like radiation. Okay, so if I took the carbon 13 isotope version of methane, natural gas, CH4, carbon 4 hydrogens, and I took the carbon 12 version of methane, the normal methane, normal natural gas, and I put a match to it, I'd blow myself up equally fast either way. It'll be a tiny, tiny bit slower with the heavier isotope, but it wouldn't be enough for me to notice. I get torn apart and flung all over the place in flames. Okay, not a good idea, right? But those heavier isotopes react just a tiny, tiny bit slower. Now, in a single chemical reaction like burning methane, that difference is insignificant. But living processes often take some kind of thing, like carbon dioxide from the atmosphere. And then they do many things, one step after another. And 8 or 10 steps later, they make something else, like taking carbon dioxide and turning it into glucose. That takes about almost a dozen steps to do that. If you use carbon 13 instead of carbon 12, each one of those steps is a little bit slower, and overall, the whole process will be slightly slower, detectably slower though. So living organisms in their biochemistry will preferentially use the lighter carbon 12 isotope because it reacts just a little tiny bit faster. So if you look at sugar that was produced, glucose that was produced by a plant instead of in a chemical factory, and you looked at it, you would find that biologically produced molecule is slightly enriched in the lighter carbon 12 isotope and slightly depleted in the carbon 13 isotope. And that difference will be enough to measure. So, carbon and some other types of molecules, some other types of atoms, carbon isotopes can be used as a potential biosignature. A flag saying this molecule was probably produced by a living organism rather than by a simple chemical reaction. Now, why is that useful in science? Well, you can make a crude, there's other factors I'm not going to go into, but you can make a crude estimation of how much biological productivity was in an environment in the distant past by seeing how much buried carbon, like oil or coal or any other kind of thing, is enriched in carbon 12 and depleted in carbon 13. So you go back 350 million years ago, what we call the Carboniferous period or the Coal Age. That's where we get most of our coal and some of our oil deposits from. You look at that, and that stuff that you dig out of the ground, that coal, is enriched in carbon 12 and depleted in carbon 13. If you look globally, you find that there is a high level of carbon 12 relative to carbon 13. It tells you that the global ecosystem was extremely productive in those days. Now go back about 650 million years ago, and you find a sharp decrease, lot less carbon-12 to carbon-13 than you found before. It's telling you that most of the carbon that got buried was inorganic carbon. In other words, it was not processed through some living organisms. And it tells you around that time, global productivity, global biological productivity took a sudden catastrophic nosedive. Reason why? Geological evidence suggests that the planet went through a glacial episode that froze the planet almost from pole to pole. Even down into areas like South Florida or Mexico, where those things are now, or maybe Central America, would have been covered in ice. So not surprisingly, if the planet almost froze over, there are a couple of those, what we call snowball Earth episodes around. Not surprisingly, when that happened, when you have these massive catastrophic ice ages, that wiped out the majority of life on this planet. And that shows up in these ratios between carbon-12 and carbon-13. So looking at these kinds of stable isotopes, carbon being one of the big ones, but they also use some others, that's giving you an indication of how much biological productivity was around even millions or hundreds of millions of years in the past. And you see a sudden change in that, it's telling you something nasty has happened. Something has changed life on this world. So that's where the geochemists come in and tie in with the paleontologists to get an understanding of the progression of life through the ages. Now, one other thing that may become relevant. We've got a spacecraft on Mars running around this crater that clearly shows signs that it was covered with water, at least periodically. They've dug up rocks and analyzed them that are described as mudstones. In other words, like basically cemented together mud. They found an old stream bed billions of years old, and they sampled rocks from there and stuff like that. That curiosity rose. Well, one of the big things is, okay, this area had water way in the past. Is it possible that some living organisms were there at one time? Well, they've got an instrument that can detect, called a mass spectrometer, that can detect the ratios between carbon-12 and carbon-13. If they find, now I haven't found it yet, and there may not be there, but they can detect organic molecules, and they can see what the carbon ratios, isotope ratios are. If they were fine organic molecules, right now they have them down to the parts per billion level, but as they analyze the data more, they'll go down to the parts per trillion level. But if they find organic molecules that are depleted in carbon-13 and enriched in carbon-12, that is a potential, not a definitive, but a potential sign that there was at one time some kind of primitive life on Mars. And then what NASA does is go up to Congress with a big tin cup in hand and say, brother, can you spare about $20 billion for a sample return mission? They'll love that, but especially this Congress. But at any rate, that's one of the purposes of that mission, to detect presence of water, and if they're extremely lucky, detect organic molecules, and if they're very, very lucky, see if the carbon-12, carbon-13 ratio suggests that those molecules were produced by living organisms sometime in the past. So we can do all kinds of cool things with various types of isotopes, and those are just a few examples. All right.